Hi, I'm Dave Morgan from the University of California in San Francisco. And in this lecture, I'm going to tell you a little bit about our work on the regulatory system that drives the cell through the metaphase to anaphase transition. The theme of this part of my lecture is going to be synchrony. And by that I mean that in biology, there are many occasions in which multiple entities, such as chromosomes or genes or cells, have to be commanded or regulated in such a way that they respond synchronously to a single input signal. And so today I'm going to talk about how the chromosomes of the mitotic cell are behaving synchronously and what the mechanisms underlying that synchrony are. So we're going to focus in particular on the metaphase to anaphase transition, this moment in the cell cycle when the cell goes from the metaphase state, where the sister chromatids are aligned on the mitotic spindle, and suddenly separates all of its sister chromatids more or less synchronously and pulls them to opposite poles of the cell. And the synchrony of anaphase is particularly well illustrated by this movie here, which shows a vertebrate cell in which the mitotic spindle is labeled in these reddish-orange colors. The microtubules are reddish-orange, and these green dots represent the kinetochores, where those microtubules are attached to the sister chromatids, and the sister chromatids themselves are visible as these dark shadows in the middle of the spindle. And so when we start this movie, what we'll see is this metaphase cell progressing into anaphase through the synchronous separation of the sister chromatids. And so right about there, the sister chromatids begin to separate and are pulled to opposite poles of the cell. And the key thing that I want you to focus on in this movie is how synchronous sister chromatid separation is. Once the sisters begin to separate, they all separate at more or less the same time. So, what I'm going to tell you about today is a little bit about the regulatory system that helps drive that synchronous sister chromatid separation. And to do that, I need to remind you about some of the basic features of cell cycle control. As is well established, it's now known that the cell cycle control system involves a series of CDK cyclin complexes that are activated at different cell cycle stages, and then that takes the cell up to metaphase here when the anaphase promoting complex, or APC, is responsible for targeting specific proteins that bring about the metaphase to anaphase transition. So the APC is the regulatory molecule that lies at the center of my talk today and lies at the center of any discussion of the metaphase to anaphase transition. Now, the APC drives anaphase through a fairly complex circuit that's illustrated on this slide here. It has two major targets, the first of which is a protein called securin. And securin is a tight binding inhibitor of a protease called separase. And separase, so that when securin is destroyed in mitosis, separase is activated, allowing it to go to the sister chromatids and cleave a specific subunit of a protein complex called cohesin that holds those sister chromatids together. And so when separase is activated, that results in the loss of sister cohesion, which allows those sisters to be separated and then pulled apart by the mitotic spindle. And so through this simple mechanism, the APC directly initiates the onset of sister chromatid separation and segregation. Now, the other major target of the APC are the cyclins, the regulatory subunits that activate the CDKs. And so when cyclins are destroyed in mid-mitosis, that leads to the inactivation of the associated CDKs, which then allows dephosphorylation of CDK substrates. That dephosphorylation of CDK substrates is essential for normal progression through the stages of late mitosis. So, for example, normal anaphase spindle behavior and the disassembly of the spindle and cytokinesis all require dephosphorylation of CDK substrates. So the question for today is, what is it about this regulatory system that drives the synchronous separation of multiple sister chromatids all at about the same time? And to appreciate one of the answers to that question that we discovered, there's one more component in this system that I need to mention. And that is a protein phosphatase that contributes to the dephosphorylation of CDK substrates in late mitosis. Now, obviously, the inactivation of a kinase alone is not sufficient to cause the dephosphorylation of a protein. You need a phosphatase to take that phosphate off. And in budding yeast, the system we work in, the major phosphatase that takes off CDK substrate phosphates is a protein called CDC14. And CDC14, this phosphatase, is activated in early anaphase at about the same time as cyclins are destroyed. And by this mechanism, by activating the phosphatase at the same time as you inactivate the kinase, that leads to a highly concerted dephosphorylation of CDK substrates. Now, what's interesting about CDC14 is that it's activated in early anaphase by a mechanism that depends on separase. In other words, through a mechanism that's really not very well understood, the activation of separase leads not only to sister separation, but also to the activation of CDC14. And so this results in a really interesting regulatory circuit in which the APC uses multiple pathways, essentially, to lead to the dephosphorylation of CDK substrates at the same time as it's causing this separation of the sister chromatids. But the question for today is, what is it about this regulatory system that drives synchronous separation of all the sister chromatids at the same time? Now, an obvious possibility is that separase activation has to assume certain characteristics that allow separase to be activated in large amounts very suddenly, so that it can spread very quickly through the cell to all the sister chromatids and basically cleave the cohesin more or less simultaneously on all those sister chromatid pairs. But there's no real indication in this system of any such sort of switch-like separase activation. In other words, one might expect that there might be positive feedback in this system somewhere that could generate a highly abrupt and complete activation of separase, which could then spread rapidly through the cell to generate sister separation in a synchronous fashion. And so I'm going to tell you today about some of our work that led to the discovery of a potential positive feedback loop in this system that helps generate rapid and steep separase activation in the yeast cell. And these studies began, innocently enough, when we were looking at the phosphorylation state of this protein here, securin. Now, securin had been identified in our earlier studies as a potential CDK substrate, something that might actually be phosphorylated by the CDKs. And it's well known to be a good CDK substrate in vitro, and so we decided to analyze its phosphorylation state in the cell using mass spectrometry. And those studies, which were carried out in collaboration with Andrew Kruczynski at UCSF, led to the identification of these 4 phosphorylation sites on securin that are all found at CDK consensus sites, SP or TP residues that are known to be CDK consensus sites. Interestingly, 3 of these sites had been identified previously by others as involved in various ways in the regulation of separase by securin, but this one site over here had not been studied in any detail, and we found this one to be interesting because it's located right next to a sequence called the D box, or destruction box, of securin. And that destruction box is known to be required for the recognition of securin by the APC. Likewise, there's another putative CDK site that we didn't identify down here next to another sequence called the Ken box. And that Ken box, likewise, like the D box, is known to be involved in recognition of securin by the APC. And so an obvious hypothesis was perhaps that phosphorylation of this serine right next to the D box could interfere with recognition of this protein by the APC. In other words, CDK-dependent phosphorylation of securin inhibits its destruction via the APC. So we tested that hypothesis by the most direct means possible, which is simply to test the ability of phosphorylation to inhibit the ubiquitination of securin in an in vitro assay using purified components. And so this gel, this is a polyacrylamide gel autoradiograph showing the ubiquitination of a radio-labeled securin protein here by purified APC in vitro. And so what we do for these reactions is produce securin by in vitro translation in a radio-labeled form, and then incubate it with purified APC and all the other components you need to get ubiquitination of a protein. And that results in this ladder of bands that forms above the securin, which represents the addition of multiple copies of this ubiquitin protein. And so in the absence of kinase, clearly securin is a good APC target, as we expect. And the question then becomes, how good a substrate is it for the APC after it's been phosphorylated? And the answer to that is shown here. Once securin is phosphorylated, its mobility on the gel shifts, but it is no longer a good substrate for the APC. Phosphorylation of securin has therefore greatly inhibited its ubiquitination by the APC, just as we predicted from the position of the phosphorylation sites. Then, of course, we were also able to restore ubiquitination of the protein by treating that phosphorylated protein with the phosphatase, CDC14. And so by adding CDC14 to the reaction, we can now restore ubiquitination of securin, indicating that this phosphoregulation is reversible by the appropriate phosphatase. So what does all this mean for the regulation of anaphase? Well, it has a number of interesting implications, the first of which is that we can now add another regulatory arrow here, suggesting that cyclin CDK activity is capable of inhibiting APC-dependent securin destruction. And this might have implications for the order of events in anaphase. It might suggest that some level of cyclin destruction is required before securin destruction can occur. But more importantly, we can also add another arrow here, suggesting that CDC14 can feedback and stimulate securin destruction. And this is interesting because securin destruction leads indirectly to CDC14 activation. In other words, what we have here is the potential for a positive feedback loop. And the idea would be, therefore, that at low levels of securin destruction, that might lead to the low levels of CDC14 activity, which could feed back and promote additional securin destruction. And one might imagine that under certain conditions, that would lead to the switch-like abrupt activation of separase in the cell. In other words, the sort of behavior we were looking for to explain the synchronous behavior of anaphase chromosomes. This next slide illustrates that same point in a more graphic fashion. What we're seeing here is that if there is no feedback in this system, a linear increase in APC activity would be expected to lead to a linear increase in separase activity. But if positive feedback exists in the system, then one might imagine that there would be an initial delay in securin destruction until some threshold level of destruction is achieved, where CDC14 positive feedback kicks into gear and essentially flips this switch from low separase activity to high separase activity. And this steep separase activity might be expected to contribute to the synchrony of anaphase chromosome behavior. So how do we test that possibility? Well, what we needed to do was disrupt this positive feedback loop specifically in an intact cell and then look to see how anaphase occurs in that cell. And to do that, we developed the securin 2A mutant, in which we've mutated the two CDK phosphorylation sites, the one that we actually identified, plus we also mutated the other site that is a CDK consensus site, in case that is also phosphorylated in vivo. And so we found that, sure enough, when we mutate these two phosphorylation sites, that abolishes the ability of CDK1 to inhibit ubiquitination of this protein. So we then expressed this protein in a yeast cell. In fact, we replaced the endogenous securin gene with the securin 2A mutant gene, thereby resulting in a yeast cell that should not contain this positive feedback loop. And then we analyzed anaphase in those cells. And the first result is that those cells appear quite normal at first glance. They grow normally on yeast plates and so on, but as you'll see, upon detailed analysis of their mitotic events, we found that they do indeed have some serious defects, as I'll describe in the next few slides. Of course, the obvious hypothesis is that expression of this securin 2A mutant might somehow reduce the synchrony of anaphase in these cells. And so then the question becomes, how do we test the synchrony of anaphase? And to explain what we did, this slide illustrates the underlying thought process that's going into the experiments I'm going to show you. Now, one might imagine that when separase activity rises in early anaphase, that will result in slightly asynchronous separation of different sister chromatid pairs, because one might imagine that different sister chromatid pairs would have different sensitivities to separase. One chromosome might separate at a lower level of separase than another. And so as a result of this, one can imagine that there should be some small amount of time between the separation of two different sister chromatid pairs, which reflects the synchrony of anaphase. However, our prediction is that in the securin 2A mutant, the activation of separase is going to be less steep than it is in the wild-type cell, and as a result, there should now be more time between the separation of two different chromosomes. In other words, a loss of synchrony will be reflected in the fact that there is more time between the separation of two different chromosomes. And so what we needed to do, of course, was measure the relative timing of separation of two different chromosomes in a yeast cell. And this is not a simple task, for the very simple reason that yeast cells are extremely small compared to the giant animal cells whose mitoses I've been showing you in my previous slides. So it's not possible to do this sort of high-resolution microscopy on a yeast cell for the simple reason that it's so tiny. And so what we needed to do was specifically label certain sister chromatid pairs in the yeast cell using fluorescent tags and then observe the timing of their separation that way. And to do that, we developed this yeast strain that began with the construction of a red fluorescent tag on the spindle poles. So using red fluorescent protein attached to a important spindle pole protein, SBC42, that resulted in the appearance of a red fluorescent tag on the spindle poles. Then more importantly, we labeled a couple of chromosomes with green fluorescent dots. So first, we started with a green fluorescent dot on chromosome 4 here. And to do that, what we do is we insert an array of lac operator repeats on the arm of chromosome 4, and then express in these cells a lac repressor that's been tagged with green fluorescent protein. And so that lac repressor, when it binds to these repeats, these lac operator repeats, results in a nice green fluorescent dot on the arm of chromosome 4. That dot will, during metaphase, will be a single dot, but when they separate in anaphase, that dot will become two dots. And so we can determine the timing of separation of chromosome 4 that way. Likewise, we labeled another chromosome, chromosome 5 as well, and in this case we used TET operator repeats and expressed in the cell a GFP tag version of the TET repressor, and that results in a green fluorescent dot on chromosome 5. And as I've indicated here, basically fortuitously, it turned out that this chromosome 5 green fluorescent dot was much brighter than the dot on chromosome 4, and that actually allowed us to distinguish between the two chromosomes in our experiments. And so what we've created is a yeast strain in which we have two different chromosomes labeled with green fluorescent dots, and now what we do is use microscopy to measure the relative timing of the separation of those two chromosomes as an indication of anaphase synchrony. And so this next slide shows what happens in a wild-type cell. And what we did for these experiments is we subjected these cells to spinning disk confocal microscopy, in which we're basically taking images every 10 seconds over a period of several minutes in an asynchronous population, and then we go back to the videos and look for mitotic cells as they progress through the metaphase to anaphase transition. And one such cell is shown here in this video montage. And so we're starting out in the upper left with a metaphase cell in which there are two green dots visible in the cell. And then at this time point right here, at the 20-second mark, that top green dot separates into two green dots, indicating that one chromosome has now separated and segregated. And then we follow this time course along every 10 seconds until at this time point right about here, the other green dot separates into two distinct green dots, and that of course represents the second sister chromatid separation event. And then from that point onward, those separated sisters then migrate to opposite poles of the anaphase spindle until by the end of anaphase here, the two green dots of each sister are now fully segregated to opposite ends of the cell. So there were a few surprises in these results. First of all, the big surprise was the amount of time between the separation of these two events. Clearly 90 seconds difference in the separation of these two sister chromatid pairs represents an awful lot of time, given that mitosis in these cells is only a few minutes long. And so we were a little surprised to discover how asynchronous anaphase actually is in these cells, not nearly as synchronous as we thought it would be. That was the first surprise. The second was that using the relative intensity of the green dots, as I said, we could distinguish between chromosome 4 and chromosome 5, and that led to the surprising realization that in all these cells, chromosome 4 is always separating before chromosome 5. In other words, chromosomes don't separate in a random order, but appear to be ordered such that 4 always goes before 5. So the big question, of course, is what does this look like in the mutant securin 2A cells? And in the next couple slides, I'll show you those results. So first, this slide gives a summary of all the results with a number of different wild-type cells that were analyzed, simply looking at the time between separation in a large number of cells. So this is a histogram showing the distribution of results in wild-type cells. And as I mentioned, the median time of separation is about 90 seconds in these wild-type cells. Then in the securin 2A mutant, sure enough, we saw a significant delay in the timing of separation of the two chromosomes, such that the median time of separation was more like 170 seconds rather than 90. And so we're seeing a roughly twofold increase in the amount of time between the separation of these two chromosomes, suggesting that sure enough, this positive feedback loop is important for the synchrony of anaphase in these cells. This also has an impact on the accuracy of chromosome segregation. By looking at the green dots in these cells and counting the number of dots in the population, we can actually get a pretty good idea of the frequency with which chromosomes are missegregated during mitosis. And by that sort of approach, we estimate that 2.5% of the cells in the securin 2A population have experienced a serious segregation error, indicating that this positive feedback loop is really important for the normal fidelity of anaphase as well. And so, in summary, the idea is that the positive feedback loop that we've identified here, this dephosphorylation of securin, is really crucial for the synchrony of anaphase and probably also for the fidelity of anaphase, and that these positive feedback loops like this one and possibly others that we haven't yet identified are important for the normal high fidelity, high synchrony anaphase that is observed in most cells. It's not clear at this point whether these sorts of regulatory mechanisms also exist in higher eukaryotes, although I think it's very likely that they do, and will be discovered in the coming years. So why is all this important? Well, the fact is that cancer, a very common disease of humans, of course, is in large part a disease that's brought about by genomic instability. And errors in chromosome segregation are likely to be very important in providing genomic instability that leads to an enhanced rate of tumorigenesis. And so understanding these detailed mechanisms of chromosome segregation is really crucial for understanding the genesis of cancer. And with that, I'd like to thank the people who did all the work that I've told you about in this lecture and in my second lecture, because none of the things I showed you were actually done by me personally. They're all done by a variety of students and wonderful collaborators with whom I've worked over the past few years. Two of the people that did much of the early work with the CDK substrates in my second lecture, Jeff Ubersachs and Mart Logue, are previous lab members who established a lot of that important early work. And then the more recent studies of CDK substrates were carried out by Liam Holt, a student who collaborated on the mass spectrometry project for the identification of recent CDK substrates. Liam was also responsible for the anaphase switch studies that I just told you about in this lecture. Throughout these studies, our collaborator Kayvon Shokat at UCSF has been instrumental in the analysis of CDK substrates. In my second lecture, I told you about how the evolution of CDK substrates might give some interesting insights into mechanisms of phosphorylation, and that was all done by a bioinformatics student, Brian Tuch, in Sandy Johnson's lab at UCSF. Kurt Thorne helped us with microscopy in this anaphase project that I just told you about, and Andrew Kuczynski is a mass spectrometrist who helped us with the phosphorylation sites on securin. And finally, Judith Villain, a postdoctoral fellow in the lab of Steve Gigi at Harvard, performed this remarkable large phosphoproteomic analysis of CDK substrates that I told you about in the second lecture. And thank you for your attention.